My name is Dmitry Sutormin, and I'm postdoctoral fellow in the Anna Kuchna laboratory at ISB. So I joined ISB in 2022, and I earned my PhD in Russia, in Moscow, for the development of a novel molecular biology technique to study a specific group of enzymes at an unprecedented scale and resolution and sensitivity. But today I will not talk much about that technique because it's were my previous achievements. So I'm really a tech guy. I like to develop novel molecular biology techniques and push the boundaries of the current microbiology using that techniques. And that's why I joined the ISB, because ISB is a great place to develop and apply novel methods thanks to the systems biology approach, which gives really good vision and opportunity to do so. So here at ISB, my challenge is to apply single-cell transcriptomics for the natural microbiomes. So the single-cell transcriptomics in bacteria, it is more or less established, but it's still a very novel method. And we routinely apply it to the laboratory strains and to the laboratory bacteria. But what about the natural microbiomes? Can I just take an environmental sample or human gut microbiome sample, something like that, and profile the diversity of bacterial physiological response with that sample? Not really, and the standard methods, they didn't work, so it required 5 rounds of optimization. It required the development of a novel software and novel analysis algorithms to leverage a completely new set and type of data. So that took more than around 2 years of optimization, but finally I was lucky to make this method work, and we named it, by the way, multi-microsplit. So I was lucky to optimize that method to the level of for the application, and I applied that method to several microbiomes obtained from the human donors. So these were human gut microbiomes. Why is that important in general to study the diversity of physiological responses in bacteria at a single cell level? Because in general, we treat bacteria as a very simple organisms. They are single cellular, they do not form tissue, they do not form bodies. They have pretty small genomes, not that many genes encoded in those genomes compared to eukaryotes. So what's the matter? It turned out that if you see physiology of individual bacterial cells just in a homogeneous environment, like in a very precisely controlled laboratory conditions, you will observe that cells are different. They all have the same genome, they all have the same genotype, no mutations, no differences at the genomic level, but you will see that they could express different genes at the same conditions. This phenomenon is called phenotypical heterogeneity. Phenotypical because it is not encoded in a genome, and heterogeneity is just reflects the difference, physiological difference across the cells. So that's what the main purpose. Why would we need this method to study the microbiomes? And because of this phenotypical heterogeneity, it has a lot of consequences. Bacterial cells, they can be affected differently by drugs, for example, antibiotics, because of the phenotypical heterogeneity. And if we can understand the mechanisms, or at least if we can describe the physiological heterogeneity, we understand the microbiomes and bacterial physiology more. So let's go back to the main story. So I applied the multi-microsplit to the human gut microbiomes, and I profiled several dozens of different unrelated bacterial species. Interestingly, I observed a common physiological pattern across all these species. It turned out that all bacterial cells, they can be classified into 3 major physiological groups. I call them guilds. The guild of stressors, and in these guilds, cells are enriched with genes associated with stress response, various types of stress responses. The second guild is a guild of metabolizers, and metabolizer cells, they're enriched with genes associated with metabolic pathways. So these cells, they are actively doing some matter transformation, some energetic metabolism. And the third group is a guild of growers. The grower cells, they are investing more into the expression of growth-related genes. So presumably, this guild of cells, they are more about replication, cell division, and population progression, something like that. And that's very interesting and novel that different species of cells, they exhibiting the same conserved pattern. Stressors, metabolizers, and growers, which means that in natural populations, bacteria are using a bed hedging strategy, which presumably helps them to survive changing conditions. Because like if the environment is good and there is a lot of supplements and nutrients here, the growers and metabolizers will be happy in this environment. So they will benefit from those conditions. But if environment has changed and there are no more nutrients here or some stress factors, given a bad time for bacteria, the stressor subpopulation will survive because these cells were prepared to survive stressful conditions. And then after the environment changes again, they will potentially give rise to a novel population of bacteria. So that's very interesting, and the method is really, really advanced. And in fact, there are only 2 laboratories in the world currently who are capable of doing microbiome scale single-cell transcriptomics in bacteria, and we are one of these 2 laboratories. But is it really important? How it is related to the human health or to the public health in general? So I'll give you one example about the antibiotic resistance. And in general, it is considered currently that antibiotic resistance, it emerges, it starts from a small group, subpopulation, of bacterial cells which are genetically identical to other cells in a population, but for some reason they develop a specific physiology so they can tolerate, they can survive antibiotic treatment. And this subpopulation of bacteria, sometimes they're called dormant cells, sometimes they're called persister cells, but in fact, they're very rare and they're really hard to find and study, but they're likely present in any bacterial population. So they are surviving, they are tolerating the course of antibiotics, and then they give rise to a novel bacterial population after the antibiotic treatment is gone, or they acquire additional mutations and they give rise a completely resistant population of bacteria, which are genetically modified or they acquired specific mutations. So they are further and more resistant to the antibiotic treatment. Going back to the guilds I just mentioned, we hypothesized that the guild of stressors, it could be functionally related to these cryptic persister cells. Because again, if a bacterial cell is, has some stress responses activated, they make it preconditioned to tolerate different stress conditions, including the treatment with antibiotic. And potentially this stressors guild, they can give rise to the antibiotic-tolerated population, which will further acquire mutations and give rise to a completely resistant bacteria. This is still work in progress, and we are going to test this hypothesis, but if it turned out to be true, then we would have patterns associated with persistent cells, because we would know which genes and stress pathways are elevated, are activated in those groups of cells. And having this list of cells, we can think about development of novel antibiotics, completely novel groups of drugs, which would target these stress pathways and inhibit them. And by doing this, we will prevent the antibiotic resistance development from the very beginning, starting from the inhibition of the persister cells formation. Again, as I just said, that's a work in progress, and we will see how it will go in the future.